I'll just give you an introduction and then we can start, right?
Sure.
So it's my pleasure to introduce today's speaker, Dr. Thomas William Seviour. So he did his bachelor's from University of Melbourne, Australia. Then his a PhD from University of Queensland, Australia. Then he started working in different parts of Australia and then UK. Then he moved to Singapore, and then right now he's an associate professor, Center for Water Technology, RS University, Denmark. His topic of research is, his topic of today's talk is G-quadruplex and extracellular RNA coexistence Pseudomonas Biofilm matrix. So I hand over the session to Thomas. So it's yours, and then you can start, Thomas.
Yes, thank you very much. So yeah, it's a pleasure to be here with you online. It's a shame I cannot be there personally. I have very fond memories of my time in Tamil Nadu, and I know some of the people in the audience, including Kumaravel and Saravanan. I see he's in the audience too. So big hello to you guys. So I'll talk today, about something that I've been working on for quite a lot of time, and I sort of started working on this in Singapore many years ago, and I've been fortunate enough to continue this work. That sort of led us into exploring extracellular DNA in biofilms, and so in doing so, continue this sort of journey of exploration on this amazing molecule that was started by these guys, Watson and Crick, many years ago. It says my mic has been disabled. Can you still hear me?
Yes, you can. It's audible. It's audible.
Okay, that's good. Fair enough. So, but my starting point here was reviewing, investigating biofilms. I mean, that's my background, and my interest is in biofilms, and Kumar mentioned that not all of you will be very familiar with biofilms. I know that some of you are incredibly familiar with biofilms, but for those that don't know, I mean, biofilms are they're the cause and cure of a range of environmental and medical problems. In the environment, they're used for many good things, like wastewater treatment, bioremediation, but they can also be the cause of very, very negative things, such as chronic infections in clinical settings. And here's an example of what can happen if there's a chronic biofilm that cannot be treated. It can lead to horrible wounds. And biofilms are maybe I should have biofilms are for those of you that don't know, they form when bacteria stick together, and they stick together by secreting an extracellular matrix. And in doing so, they cease to be free-living, planktonic, free-spirited bacteria that's swimming around freely in liquids, but they stick together and they adhere to surfaces, and they adhere to each other. And this has a number of consequences, and one of the consequences is that they behave very differently in these biofilms. The biofilms enable all these complex relationships to emerge. You know, the bacteria are able to communicate, cooperate, compete. So another interesting aspect of biofilms is that the bacteria in biofilms become very, very resistant to environmental stresses. And one of those environmental stresses or stresses that they can become resistant to, of course, is antimicrobial stress, and this can result in increasing antimicrobial resistance. So understanding, you know, the molecular basis of why biofilms form is potentially a key part of identifying strategies to fight rising antimicrobial resistance. And so the question is, why do these biofilms form? And I mentioned that they secrete these extracellular polymers. And these extracellular polymers can be a range of different polymers. They can be polysaccharides, they can be proteins, and more recently, our focus, or my focus, has shifted towards these extracellular nucleic acids. But one of the reasons that it's so difficult to get to the bottom of what biopolymer is present and what they're doing and how we can control their secretion and their function is because individually these biopolymers are very difficult to characterize. They're very complex. They're very poorly soluble. So it's very difficult, very tough research, sort of getting to the bottom of precisely which biopolymer is secreted and why and what it's doing. But it's important. As I said, we want to get to the bottom of what biopolymers are present in biofilms. I've described this previously as an identity crisis, because if we think about, I'm not sure what the background of the audience is, but microbiologists are very familiar with the composition, the structure of cells. We understand that there's a nucleoid where the DNA is packaged, and that they have these membranes, phospholipid bilayers, and et cetera, et cetera. So the organization and assembly of the cells is very well understood, but outside the cell is very poorly understood. But if we do understand that, we can, as I said, perhaps better improve some of these horrible biofilm-associated infections, and also perhaps we can improve bioprocess controls in the settings where biofilms are beneficial, such as in wastewater treatment or bioremediation. But this is my challenge for a long time, was how do we really understand which biopolymers are present, and the classic approach, the classic approaches to assigning function to biopolymer in biological systems or bacterial systems specifically just didn't work for biofilms. This was using genetic knockouts or tagging particular proteins and what have you. It just was not effective for biofilms, because we can't do some of the sort of classic controls that are necessary, like add backs and what have you, and it's difficult to do these things when we don't really have the most sort of basic understanding of what's going on. So my role, or what I started doing, was very old school sort of a approach of and we can see it here, of taking a biofilm with lots of cells and trying to dissolve it, get rid of the bacteria, get rid of everything that was not contributing to the formation of this extracellular matrix, which is what we're able to do then, and then try to understand the molecular basis of this matrix. That's essentially what we did. And to give you some insight into what this matrix is like and some of the properties in the matrix that we're interested in, this is a video that shows it's actually, this circle here is a bacterial-sized magnetic bead that's immobilized in a Pseudomonas biofilm, and when we applied a molecular force, we see that this bacteria is attracted to a magnetic force. You see that the bacteria is attracted to the magnetic force, and then we remove the magnetic force, it returns to its original position. So that's telling us that it's like a tennis ball. It's storing its energy and then it's releasing its energy, which tells us that Pseudomonas biofilms are viscoelastic gels, and they're viscoelastic gels because they have this three-dimensional cross-linked polymeric network, and I'll show you what that looks like. So when you deform the biofilm and then remove the force, the magnetic force, it returns to its original position. So it's like a tennis ball, pressing a tennis ball, removing a tennis ball, and then it returns to its original position because it has this network. And so what I sought to find out was what is it? What is a constituent that is contributing to this network forming? And yeah, I guess first I should say that Pseudomonas aeruginosa is a priority pathogen, and it's a lot of the images that I showed earlier of this green horrible necrotic infection, a lot of that is Pseudomonas. It's one of the ones that we're worried about with regards to antimicrobial resistance and what have you. So so that's why we looked in Pseudomonas. Yeah. But so I wanted to understand what it was that was causing this incredible behavior. And I, I'm looking at bacterial systems, but I'm actually chemical engineering, and a chemical engineering approach is brute force, dissolve everything, and then try to recover a gel. So that's what I tried to do. And the strategy for dissolving Pseudomonas aeruginosa biofilms was to put them in sodium hydroxide or sodium bicarbonate, or elevating the pH, and that was understood to dissolve these biofilms, and it's very, very effective. But as a chemical engineer, if it's a gel, which is as suggested by its behavior, then you would expect the pH transition to be reversible, but it was not. And in the case of polysaccharides, for example, that cause gels, it is. And to this point, it was believed that the network in Pseudomonas biofilms was due to one of a number of polysaccharides, either PEL, PSL, or alginate. So this observation that I could not reverse the pH transition sort of made me think, well, maybe it's not a polysaccharide. And because I could not reverse that transition, similarly, I was not able to recover a polysaccharide when I tried to isolate or fractionate the biofilm after I increased the pH. So the question was, what makes Pseudomonas biofilms viscoelastic? So we had this model biofilm that we did all our studies on, which ended up looking something like this, or something like this, where we grew up Pseudomonas in a static culture. We watched it, and it formed these sort of really sticky masses, either on the surface. These are on pellicles, and this is the wild type biofilm. And what we found was that these biofilms displayed viscoelastic behavior, as suggested by the previous test using these magnetic beads, and that's shown here by the fact that these solid shapes are an order of magnitude greater than these closed shapes. And you see it for the stars, and you see it for the circles. So it's a viscoelastic solid. But when we dissolved the biofilm with DNAs targeting the DNA, the hydrolytic enzyme degrading DNA, then these biofilms degraded. So then we sort of thought, well, maybe DNA is the primary structural component. And then, but we still needed to be able to, in order to say anything meaningful about how DNA assembles into these gels, we still needed to be able to reconstitute the matrix into a cell-free, what is essentially a cell-free or DNA-only gel that behaved like the biofilm. And so we couldn't do that using alkalization. So I mean, as an engineer, I sort of looked at different solvents that could do this, and eventually found this family of solvents called ionic liquids. I'm not sure whether any of you know what ionic liquids are, but these are salts, these are salts that are liquid at room temperature. So they're incredibly effective at disrupting hydrogen bond interactions between biopolymers. And so I was able to use this ionic liquid to dissolve these biofilms and then purify them through multi-stages of precipitation, and then reassemble the DNA into what was effectively a cell-free biofilm. And this is what it looked like after doing that process. I don't know whether you can see it, but on the bottom right-hand corner, you can see that gel, that transparent gel film. So I was able to take something quite dirty, and so take something quite dirty here, using this isolation procedure, converted into something that was very, very pure, consisting and very, very pure and gel-like, with the same behavior or properties that we observed in the biofilm, but this consisted purely of DNA. And importantly, it behaved, its biophysical signatures were the same, exactly the same as the biofilm from which is extracted. And by that, I mean the temperature at which it dissolved, and which is about 57 degrees, and the pH at which it dissolved, which was, you know, alkaline pH. So what that told us, what that told me, is that this gel consisting of eDNA is assembled the same way when it's isolated as it was in the biofilm. So that was fantastic. So then we could really start understanding or addressing the question of how the eDNA becomes viscoelastic. And the reason that any biopolymer can display viscoelastic properties is because it can form cross-linked networks. So this observation that eDNA was forming these gels, that eDNA was supporting the formation of these viscoelastic networks or scaffolds in these biofilms, told us that the DNA was forming cross-linked networks. And this is not what we understand DNA to do. We're more familiar with DNA as this genetic template, you know, that exists in the nucleoid of cells as this sort of super amorphous, super coiled structure. So then the question was, how does DNA undergo this transition from supercoiled chromosomal DNA to this viscoelastic extracellular network in biofilm matrices? But the good news was that we could assemble it, we could extract it, we could assemble it into a cell-free or pure biofilm eDNA gel. And that was critical, because then we were able to understand how these nucleobases, how the nucleobases were assembled. So we, you know, as suggested by the first page in this slide in this presentation, showing Watson and Crick, these were the guys that identified DNA as a genetic code and identified that there was a particular pairing of nucleobases, two particular pairings of nucleobases that are A to T and G to C. I mean, that's the basis of you know, the central dogma of biology. That these bases that G will preferentially complement with C, etc., and T with A. And given that we could get a gel, what I was able to do then was produce a gel where the nitrogens were labeled with this radioactive isotope 15N, and then I was able to observe the nature of the base pairing in the DNA, extracellular DNA of these biofilms. And I was able to do that using solid state DNA, because these are gels, which means that we can observe solids. And using solid state NMR, I was able to look at the correlate, whether there are any correlations between nitrogens and protons, because these nitrogen proton interactions are the backbone or the basis of the G to C and A to T coupling. And what I saw, not surprising, was I saw this G to C coupling of this. So in this particular coupling, there are two nitrogen and proton interactions. There's the bonded nitrogen and proton interaction, which is red, and that's what I see here. And I also saw the through space nitrogen to proton coupling with a cytosine, which is this one here. And I also saw it for thymine to adenosine. But what I also saw were these blue cross peaks. And these correlated with the shift that you expect from a guanosine amine that is coupling with this oxygen of a guanine. So this is what's called a non-Watson-Crick or a non-canonical, or in fact a Hoogsteen G to G base pair interaction. And I also saw it for thymine, this T to T base pair interactions. And then I looked for them in the chromosomal DNA of the same bacterium of Pseudomonas aeruginosa. So this is in the biofilm, and this is in the chromosomal DNA, and I didn't see them. I didn't see the G to G or the T to T, these non-canonical base paired interactions. So these non-canonical G to G or T to T base paired interactions were characteristic of the extracellular DNA from these biofilms. Also, I mentioned earlier that these biofilms had characteristic biophysical signatures in that they dissolved at a particular temperature, which is 57 degrees, and they dissolved at a particular pH, which is pH 13. And what we also observed was that when we heated the extracellular DNA, this one here, when we heated up to 57 degrees, where it lost its viscoelasticity, at the same temperature, it lost these non-canonical G to G, T to T base paired interactions. So that told us that these G to G and T to T, these non-canonical base paired interactions were actually characteristic of eDNA, and that they were likely contributing or involved in this ability of the eDNA to form viscoelastic networks. But then, remember, we observed this only in this cell-free extract. We wanted to see whether we could also see these G to Gs in or non-canonical base pair interactions in the biofilms itself. Now, what I probably should have included here is information that one of the forms of G to G, one of the structures, the most important structures biologically for these G to G base paired interactions is what's called G-quadruplexes. And that's when, instead of getting this double-stranded helical DNA that we all know from Watson-Crick, when these nucleobases form these G to G base pairs, what they can do is assemble into G tetrads. So you have 4 G structures or guanosine structures assembling in a tetrad around a stabilizing cation, which is most typically potassium. And when you get multiple stacks, yes, you get these quadruplexes, so 4-stranded DNA structures. And there are actually antibodies specific for these DNA structures. And so when we wanted to observe whether biofilms had these G-quadruplex structures, we were able to use these G-quadruplex antibodies. And what we observed was that when we did a propydium iodide stain, which stains DNA, yes, we saw DNA fibers throughout the biofilm. And then when we did the G-quadruplex antibody stain, we also saw that there were antibodies, and there were G-quadruplexes along these viscoelastic eDNA fibers, and as you can see here, overlapping with the eDNA fibers, these G-quadruplexes are a part of these eDNA fibers. Also, as I mentioned, when the fact that these biofilms dissolve at a high temperature at 57 degrees and plus, what we saw when we started heating them to 60 and 70 degrees, we saw that these G-quadruplexes disappeared. We also saw these G-quadruplexes disappear when we treated these biofilms with DNAs. So therefore, that told us that the viscoelastic behavior of these Pseudomonas biofilms was characterized by the presence and perhaps enabled by the presence of G-quadruplex nucleic acids. So that was, but then the question is, why do these G-quadruplex structures form? And the reality is, if you can do an in silico assessment of the tendency of DNA to form G-quadruplexes, there's a lot of software out there predicting, whether particular sequences in your genome will form DNA quadruplexes or G-quadruplexes. And Pseudomonas aeruginosa, it's a high GC content organism. Actually, it has hundreds of regions that are likely that with this tendency to form G-quadruplexes, but actually, what is more likely even than DNA to form G-quadruplexes is RNA. So we wanted to understand whether RNA was present in the biofilm, and so if you remember, if you alkalize this biofilm. This slide's a bit corrupted for some reason, but if you alkalize the biofilm, it dissolves. And so remember we had this cell-free isolate of eDNA, and we wanted to observe whether we had RNA present in it as well. So we alkalized it and then looked at solution state NMR, and then we looked at solution state NMR. Where are we? Ignore these letters. Hang on, I think they come in a. Yeah, I think they come in later, but yeah, sorry, yeah. I need to go back here. Sorry, yeah. Anyway, so what we observed was two signals in the NMR. We saw these very, for some reason my mouse is very slow to respond here. Anyway, we saw these ones here, which were, so we saw the ones in the square, which were very sort of compact signals, and if you count them, there are 8 of them. And then, but we also saw these broad peaks outside of the square. What we realized after a lot of analysis was that the eight compact, tight, small circles in that square, they result from 2' and 3' monoribonucleotides, and the broad signal was from the DNA. And so we therefore knew that we also had RNA present in these biofilms, in the extracellular matrix of the biofilms, as well as the DNA. What we know is that, the other thing I should have mentioned, sorry, was that these monoribonucleotides appeared only after we alkalized the biofilm, and which is interesting because we alkalized the biofilm at a pH that does not degrade DNA, but is known to degrade RNA. So so from this we concluded that RNA, not only was RNA present, but the dissolution of Pseudomonas biofilms that we observe upon alkalization is because the RNA is hydrolyzed or transesterified into these monoribonucleotides. So that suggested to us that the RNA was also a very important structural component. And this loss of DNA fiber structure that we see was ever, that is observed upon alkalization of or transesterification of the RNA can be observed here. So at pH 7, when the RNA is intact, we have the fibers. Then at pH 13, when we transesterify the RNA, the biofilm loses its fibrous structure. So of course, we wanted to know whether there were any particular RNA that were contributing to this, and in fact, we found a handful of RNA that were high. Well, we found quite a few, but there were some RNA that were markedly higher in abundance in the matrix than they were within the cells, and these were LAS-B. I mean, the most notable one there was LAS-B. That was consistently higher in the matrix than it was in the cell, and there are a range of others. And there are also some RNA or messenger RNAs that were more highly abundant within the cells than outside the cells, such as this CRCZ and SSRA at the bottom right-hand side there. So we had some that were highly expressed in the matrix, such as LAS-B and these other 3, and then we had some that were highly expressed inside the cell and not so highly expressed outside the cell. So what we then did, we wanted to observe these transcripts that were highly expressed in the matrix. So we wanted to see whether LAS-B, which is very highly expressed, whether we could see it in the matrix. And so we developed a method for doing this, which was called single molecule FISH, or single molecule ISH, it was in fact, which is single molecule in situ hybridization, where we developed an oligoribonucleotide probe that was specific for LAS-B, and you actually need, and each of these probes was fluorescently tagged, and you actually need 20 of these fluorescent probes in order to observe something by confocal microscopy. So what we're able to do is generate a lot of these probes for each LASB monoribonucleotide. So we're effectively able to load up each individual LASB molecule with these fluorophores to enable them to be seen. But despite that, initially we didn't see any LASB. And then we thought, well, maybe, given that we know that DNA is also there, and we know that RNA is contributing to this ability of the eDNA to form fibers, maybe the RNA is bound to the DNA, and maybe that could be why we don't see the LASB oligonucleotide. So we digested, did a mild pre-treatment with DNAase, and at a very low concentration. Then we increased the concentration. As you can see, with increasing DNAase concentration, the signal resulting from this LASP probe gets brighter and brighter and brighter. And we can see that here. So the amount of LASB that we can see gets higher and higher and higher and higher. So there was a DNAase dependent, you can see it down there, in fact. So there was a DNAase dependent increase in LASB signal. And this suggested to us that, that the LASB was therefore bound to the LASB DNA RNA was therefore bound to DNA in a DNA RNA hybrid structure. If you remember, there were also RNA transcripts that were less highly expressed in the matrix compared to within the cells. And when we did the same procedure, we only saw those transcripts inside the cell. We didn't actually see them in the matrix. Some of these transcripts are actually more highly expressed than, for example, LASB. So the fact that we only saw very specific transcripts in the matrix, only very specific transcripts in the matrix indicated to us that the secretion of RNA into the matrix was not a random event, because it didn't reflect the total transcript abundance, but that in fact it was probably a regulated event. And as here, the appearance and detection of extracellular RNA in the biofilm was no accident, because, as I mentioned, some of these other ones that were in fact more abundant overall than LASB, such as SSRA, we only see them here in the cells, as you can see in the yellow figures here, and for CRCZ as well. So we don't actually see them in the biofilm. So the appearance and detection, so which eRNA that appear in the biofilm as hybrid structures with the DNA is no accident. Furthermore, our method for assigning intra and extracellular RNA was validated by this microscopic approach, and this had not been done before. The next thing to do was to, we had observed this in our clinical biofilm, and we wanted to understand whether it was also the case in, we'd observed it in our laboratory biofilm model. We also wanted to understand whether this applied to clinical biofilms, and we were able to get some sputum from a lung patient that tested positive to Pseudomonas aeruginosa. And what we observed, we also were able to observe these LASB transcripts in the eDNA fibers network that we observed in the sputum of the Pseudomonas aeruginosa positive lung patient. And the intensity of this signal also increased in proportion to these low concentrations of DNAases that we're adding. Now, I should probably also note or mention that these were quite low concentrations of DNAase, and we didn't, they were sufficiently low that they were not degrading the biofilm structure themselves. So this observation that eRNA is bound to viscoelastic eDNA fibers also was a feature of these clinical Pseudomonas aeruginosa biofilms. And the other observation that we had was that what we also wanted to ascertain was whether or not RNAs was actually contributing to the viscoelastic behavior of these biofilms. So we treated the biofilms with RNase A, and we can see this down here in the X's, and I'll just quickly explain this graph to you. This is a rheogram. So when tan delta is 1 and lower, it means that the particular material is viscoelastic. So that's an indication of cross-linking. And so what we can see for the biofilm here in the buffer solution is the square. So it's getting down to 0.1. So that tells us that it's very viscoelastic. When we treated it with RNase A, which is targeting these RNA structures that I mentioned before, actually, we only saw a very, very slight increase in tan delta. So it shifted slightly closer to 1, so it was less viscoelastic, but it still was a viscoelastic structure. So we thought, what if it's RNase hybrid structures that we should be targeting, which are these DNA RNA structures? And so we treated with RNase H, and that was more effective. It shifted it even closer to this figure of 1, but it was still a viscoelastic structure. And finally, we coupled this mild DNase treatment that we described previously of 0.1 milligram per mil. We coupled that with RNase H, and we were able to completely dissolve the biofilm. So when we just have the DNase-1 and the RNase buffer, that was actually that was our control there. So DNase H itself didn't degrade the biofilm, but when we coupled it with RNase H, it completely dissolved the biofilm. So that told us that these DNA RNA hybrid structures contribute to the viscoelasticity of biofilms. So in conclusion, these G-quadruplex structures were seen to be key and characteristic of biofilm viscoelasticity. Extracellular RNA is also present and also appears to be key to viscoelastic networks. eRNA is bound to eDNA in these hybrid structures, and the release of eRNA into the eDNA fibers might be regulated rather than a random process. And we think that the presence of RNA there is crucial because DNA is a relatively boring molecule, whereas DNA has this incredible structural flexibility. As we know, it can form hairpin structures. It can exist as messenger RNA, of course, and it has this far greater propensity to form quadruplex structures. So the fact that RNA is there, we think, is crucial in the ability of these DNA structures to form G-quadruplexes. And that's brings me to an end of this presentation. What I will say is that we have continued this work, and unfortunately I cannot share that work with you because that's sort of subject to we've submitted that work for patent approval. So unfortunately, I cannot share that work. But hopefully, if I have another opportunity to present to you in 6 months' time or something like that, I can give you an update on the follow-up from this work, where we actually now understand and have the molecular basis for how this is regulated. But that unfortunately that will have to be another time. But thank you for your attention. Very happy to field questions.
Yeah, thanks, Tommy. It's pretty great. It's a pretty good talk. So there are some questions in the chat. You could just have a look at it.
Yep. Let me have a look at the chat. Okay. So G-quadruplex, I've got a question here from Dr. Sivakumar. G-quadruplex is G-rich, yeah. In this case, how is the quadruplex, how specific is the quadruplex for antibody binding? Is that the question? Look, that's a good question. Actually, what what I should also have said is that this finding, this observation of G-quadruplex, I mean, this was the first step really in our journey. Was, you know, we saw these G-quadruplexes that led us to looking into these RNAs, and now, you know, we subsequently have a mechanism, but this observation of G-quadruplexes in biofilms has subsequently been verified by others, by multiple researchers. It's they've been found in I think Staphylococcus and various other biofilms, and it's believed that they are actually very, very common structures, not just in Pseudomonas biofilms. We did look at the specificity of the binding site is the G-quadruplex itself. So this four-stranded tetrad, and we validated the specificity of this and demonstrated that it didn't bind to double-stranded DNA. But as to you know, the molecular explanation of its binding site, I don't know. But the specificity of binding has been verified. This is a commercially available antibody. How do G-quadruplexes influence gene expression in Pseudomonas aeruginosa biofilms, particularly genes associated with biofilm formation, antibiotic? So this is Dr. Gopalakrishnan. Yeah, particularly genes associated with biofilm formation, antibiotic resistance, and virulence. So yeah, G-quadruplexes, I mean, G-quadruplex actually in and of itself is a massive research field, and people are appreciating more and more that, as I mentioned, there are actually hundreds and hundreds of regions in the Pseudomonas aeruginosa biofilm that can form G-quadruplexes. As I said, it's been observed. They've also been observed in Staphylococcus and other biofilms, which are not high GC organisms. So, but G-quadruplexes are. It's understood that they, you know, that they form and they contribute to gene regulation more broadly speaking, not just for biofilm formation. What I will say is that I looked at which genes in the Pseudomonas genome were forming, you know, had this propensity to form G-quadruplexes, and there wasn't an obvious trend. I mean, what I was looking for was whether or not LAS-B, for example, had a high GC content or anything like that, but it didn't. So the genes that we saw, the transcripts that we saw, were not necessarily associated with GC-rich regions or regions of the genome with this high tendency to form G-quadruplexes. So, but it's very complex. Are there strain-specific variations? Strain-specific variations of guanine that form G-quadruplex and G. Yeah. So, I saw the other thing we did a LAS-B knockout. So LAS-B was, I mean, I didn't include it here, but LAS-B was actually very highly regulated relative to other biofilms. I mean, it stood out as being highly regulated in Pseudomonas biofilms in the extracellular matrix of the biofilms compared to other transcripts. We observed these other transcripts in the matrix of Pseudomonas biofilms, but the signal was not as bright as it was for LAS-B. So that was, it was very notable that LAS-B was more highly expressed. But of course, you know, when we did the LAS-B knockout, it still formed biofilms. So it's not so simple. And I think the conclusion we came to was that you know, one of the advantages to Pseudomonas of using mRNA is that you know, there's a lot of redundancy there. So that you know, if they lose one, then there are many a lot of other options for providing the mRNA to help the biofilm produce its matrix. And we did actually, interestingly, when we knocked out the LAS-B or looked at the LAS-B knockout, we had quite a different profile of transcripts. So yeah, so we think that LASB in this particular biofilm, yes, but LASB itself is not sufficient. And question 4: can targeting G-quadruplexes in Pseudomonas enhance the effectiveness of antibiotics or other antimicrobial agents? And that's a really interesting question. I mean, parallel to us, there's been we're not the only ones looking at the matrix. There's another group in the United States. There's a guy called Stephen Goodman. He published a paper on, I think he published a paper in Cell, I think in 2021 or something like that, but describing that there are these other DNA structures in Pseudomonas biofilms. Well, no, actually, they were not Pseudomonas biofilms. He was looking at non-transmissible Haemophilus influenza biofilms, and yeah, I think even the E. coli biofilms. But anyway, he described these Z-DNA structures, which are alternative forms of DNA structures, and they had an antibody that was specific for Z-DNA structures. And I think it's coming back to an earlier question, how these antibodies can achieve specificity in these DNA structures is I don't understand. I don't think many people do understand, but there is an antibody for Z-DNA, and what they're able to show is that by providing this antibody, they were able to inhibit biofilm formation. So that's a good question. If we added a G-quadruplex, for example, an antibody that was specific to G-quadruplexes, would it inhibit these biofilms? And we did that, and it didn't have an effect. And so that could tell us one or two things. It could tell us that G-quadruplexes are not there, and we think they are. Or what it most likely tells us is that these G-quadruplexes form inside the cells, and so they form inside the cells before they're secreted. And so by providing these antibodies, they're just not getting to the G-quadruple. And once they're formed, then these G-quadruplexes don't. It's not easy to they're quite robust, resilient structures. But it is a great suggestion, and it's certainly what I think is a really promising area of research is therapeutic. So if we're thinking about treatments for these biofilms, I think generating structures or generating agents that target specific DNA structures, I think is a really sort of promising area of research. And the other interesting thing is, and when we describe the fact that we have RNA in the biofilms, I mean, there's a certain degree of there, because RNA is not a very stable molecule. I mean, it's very sensitive, of course, to RNases. It's very sensitive to oxidizing conditions and alkali solvents, for example. But we do show in these studies that it's bound to DNA. We show that it's protected from RNAs by this binding of DNA. So we think that the fact that it's bound to the DNA does protect the RNA from degradation. Saravanan, good to hear from you. Lovely. Look forward to catching up. How does the eRNA regulate in biofilm? So that's eRNA regulate in biofilm matrix components, for example, various types of exopolysaccharides, alginates, or PSL, PL, or other public good components. I mean, that's a really, really good question, because we and you're, of course, sort of very well qualified to talk about how Pseudomonas fits in community biofilms, for example. When with these polysaccharides, alginate, PL, PSL, I mean, they're they have obvious synthesis pathways. You know, there's a PL biosynthesis pathway, PSL, and you know, in theory there are knockouts for these, but RNA doesn't. You know, extracellular DNA, there's no knockout for eDNA production in Pseudomonas biofilms. So the regulatory network, or the pathway for eDNA production secretion in Pseudomonas is not understood. With subsequent work, you know, we believe we're at a stage where we can describe why that happens, sort of from a regulatory point of view, but it's not as straightforward as when you're looking at sort of polysaccharide synthesis genes. But it's true. If you know, eDNA is something that all bacteria potentially can produce, as with eRNA. So in a community biofilm, there is scope, therefore, other bacteria to contribute to matrix building, and we have not we haven't addressed that question, but what this work has shown is that we can actually observe individual transcripts in the matrix. So we can observe, for example, LASB from Pseudomonas in the matrix, and no one else has ever been able to observe individual genes or transcripts in, you know, people have described eDNA in biofilms, but no one has been able to observe individual genes or transcripts. So it is possible, therefore, if we had an integrated community biofilm, then it would be possible, for example, to see whether the Pseudomonas transcripts, for example, are used or taken up by matrix assembly by some of these other bacteria. But it's not something that we've done, and you know, the person to address that question, or that would be ideal to work with to address that question, would be you, Saravanan, of course. Any more work? What would be the possible adjuvants that can be tagged that show synergistic? This is from Dr. Gopalakrishnan. It could show synergistic effect in enhancing the effect of inhibiting the various virulence genes in quorum sensing. Yeah, I'm not sure. I mean, our line of thinking is that I don't understand the role of, you know, I don't understand the role. We don't understand the role of quorum sensing yet in mediating this process. I mean, what we are looking into is sort of various stress factors that contribute to this, various stress responses that are contributing to this. So we haven't observed a link between quorum sensing, but we have with stress, various stress responses, and you know, and that's a problem, because if you're inducing stress, you know, most biofilm control strategies involve stressing the bacteria, but if that activates eDNA secretion systems, then that's a challenge. So I think one strategy would be to you know, if we can identify how to target the matrix with you know, target some of these important structures like G-quadruplexes in order to disrupt biofilm formation, then we can inhibit bacterial growth by stressing them without having this secondary complication of matrix or biofilm matrix production. But yeah, it's a good question.
We'll take last question. Like, I think it's it's been 1 hour. Like you have some work, it seems, is that right? Like you need to leave by 45 minutes, is that right?
Sorry, can you say that again?
So you need to leave by 45 minutes, is that right? What, like,
yeah, I have to leave. Yeah, that's true. I have to leave in a couple of minutes. That's true. Yep.
Okay.
So I need to wrap up. Yeah. That's true.
Right.
Yep.
So next question. We can go to the next one. Like there are some questions from.
Okay.
Are there any eRNA information available in mixed species biofilm?
No, well, we haven't done those. We haven't done those experiments yet. But as I said, if you know, if we had a model mixed microbial biofilm involving Pseudomonas, then we'd be able to look for Pseudomonas aeruginosa. We'll be able to look for Pseudomonas transcripts in other parts of the biofilm. But what I will say, yeah, that we had that clinical biofilm, which was a Pseudomonas aeruginosa positive lung patient, and that was a mixed species biofilm. We saw the LASB transcript in that. So we know that extracellular RNA does appear in mixed species biofilm. But as to this question of common goods and all the rest, we don't know yet whether LASB from Pseudomonas is able to be used by eDNA produced by Staph epidermidis, for example. But that's an interesting question.
So the last question will be from Gopalakrishnan. How do G-quadruplex in biofilm related exRNA modulate the interaction between Pseudomonas aeruginosa biofilm and host immune system? The last one.
Yeah, I mean, it's a good question, and as I said, we are investigating, exploring the possibility that stress induces eDNA secretion, and we know that LASB is part of the stress response of bacteria. So the stress response of Pseudomonas aeruginosa. So that's as I said. I think it's I mean, G-quadruplexes, they're more resilient. They're than double-stranded structures. So I imagine that they help in the resilience. They're more difficult to degrade, for example, than double-stranded DNA. So yeah, but if we can inhibit the stress response while still exposing bacteria to antimicrobial agents, then we have a much better chance of killing the biofilms without leading to problems with antimicrobial resistance and tolerance. Yeah.
Yeah. Thank you, Thomas. Thanks for your information. Thanks for your wonderful speech and your time to come.
No worries. Thank you. Lovely to be involved, even if it is remotely. And sorry I can't be there personally. But hopefully, hopefully in 6 months' time, 12 months' time, if you want an update on this, I'll be in a position to give you some exciting updates on this work.
Sure, we will disturb you. We'll disturb you in 6 months or 12 months' time. Possibly we'll call, come here, give a talk, see these snakes, scorpions.
Yeah, love to do that. Yeah. Okay. And eat some wild honey as well. Okay. All right, guys. Enjoy your session. Bye-bye.
Thank you.